Good morning, children. I will continue with the chapter of absorption by roots, and today I will take the topic root pressure. The root pressure is the pressure developed in the roots due to continued inward movement of water through cell to cell osmosis, which helps in the ascent of cell sap upward through the stem. Now I'll show you the figure. This is the internal structure, diagrammatic cross-section of part of root showing arrows, the cell-to-cell conduction of water from root here to the xylem. Now this root pressure is built up due to cell-to-cell osmosis in the root tissue. See this figure? As one turgid cell presses the next, the root hair cell will absorb water. The water will pass to the adjoining next cell. So this cell becomes turgid. So this turgid cell presses the next cell. The force of flow of water increases inward. When water reaches the xylem vessel, centrally placed vertical channels, the thick-walled vertical channels, it enters the pores of their thick walls with considerable force. Thus, the root pressure is one of the forces to raise up water through the stem into the leaves. But this force alone cannot push up water to the top of tall tree up to certain height. The water is being lifted up by the root pressure. But in very tall trees, along with the root pressure, one more force acts on it, that is transpiration pull. That I explained you in transpiration also. So both these forces will be responsible to take up water from the root hair cell up to the top height of the tall trees. So see the figure, the water being absorbed by the root hair cells, and then the water is flowing to the next cell, then to the third, then fourth, and so on. It will move to the internal part of root into the xylem vessels. These are the xylem cells, thick-walled cells. These xylem cells are vertical channels, centrally placed vertical channels, which in turn are connected to the vertical channels, that is the xylem vessels of the stem. And thus upliftment of water takes place due to the pressure of the root. Now we'll demonstrate and measure root pressure with the help of an apparatus called manometer. We'll take a potted plant of a balsam or tomato or bryophyllum that has been well watered the previous day. Now we'll cut off its stem 5 to 8 centimeter above the soil and immediately fix a glass tubing to it by means of rubber connection. Can you see this? This is a potted plant well watered. The stem is being cut 5 to 8 centimeters above the pot, and then it is fixed to a glass tubing by the rubber tube. Then we'll connect a manometer into the glass tubing. Observation: water will start coming out of the cut end of the stem and start rising in the glass tubing. So because of root pressure, water will come out from the cut end of stem. This coming out of water from the cut end of stem is also called bleeding. So this will push up, water will be pushed up and it will rise in the glass tubing. The rising water will raise the mercury level which is filled in the connected manometer. This is the mercury level. This is its initial level. So the mercury level will be pushed up because of the water behind is coming with a force. As a result, the water rises in the connected tube due to heavy pressure from the roots, which is called the root pressure. The rising water will raise the mercury level in the manometer. So we find the root pressure helps in upliftment of water up to the stem to certain height. Then there are some examples which we experience due to root pressure. That is, loss of water through the cut or injured stem. Bleeding is also due to root pressure. Then in early morning, drops of water along the leaf margins in certain plants such as tomato, grass and banana is due to excessive root pressure. This loss of water as water droplets from the margins of the leaf due to excessive root pressure is called guttation. So guttation and bleeding, the cause of guttation and bleeding is the root pressure. Now I will be taking the next phenomenon, plasmolysis and deplasmolysis. What is plasmolysis? It is shrinkage of cytoplasm of a plant cell from its cell wall under the influence of hypertonic solution is called plasmolysis. Now, if we are going to place any part of the plant in a hypertonic solution, that is the concentrated solution, then we'll find the water from the cell sap will move out and the cell is in the state, it shrinks, is said to be limp or flaccid, and this condition is called flaccidity, and this is due to the phenomenon plasmolysis. And flaccidity is reverse of turgidity. Turgidity is cell fully swollen and fully charged with water, and flaccidity is the shrinkage of the cell due to loss of water from the cell. Now, when a plant cell is kept in hypertonic solution, the cytoplasm of cell along with its cell membrane separates from the cell wall. See the diagram. This is the plant cell. We have placed it out. The outside is hypertonic solution. So after few hours, you will find that due to exosmosis, the protoplasmic mass, it shrinks and gets collected somewhere in the center of the cell. It gets concentrated in the center or towards one side. The central vacuole diminishes. The cell is said to be plasmolysed, and the phenomenon is called plasmolysis. The process of plasmolysis occurs because of exosmosis. The water diffuses out of the cell because of higher osmotic pressure of hypertonic solution outside the cell. I told you, if the solution is hypertonic, it means solute molecules are more in that compared to solvent. More the solute molecules, more will be the osmotic pressure and more will be the osmosis. On the other hand, when a plasmolysed cell, this cell which is flaccid now, it is a plasmolysed cell, is kept in distilled water or hypotonic solution immediately before the cell is dead. Water diffuses into the cell because of endosmosis. Now the plasmolysed cell will be immediately transferred into a container containing hypotonic solution, that is more of solvent, more of water in it. So endosmosis will take place. Water from the hypotonic solution will enter into the plasmolysed cell. The cytoplasm again gets inflated and exerts pressure against the cell wall. The central vacuole reappears, and this phenomenon is called deplasmolysis. So, deplasmolysis is the re-entry of water in a plasmolysed cell. So, that is what is deplasmolysis. Now, we will come to I'll show you the figure. This was the original cell placed in hypertonic solution. It becomes plasmolysed, and this plasmolysed cell, when immediately is placed in hypotonic solution, it will regain its original state. That is, it will become turgid again. So this is how the plasmolysis, after plasmolysis, the re-entry of water that takes place and the recovery of reversal of plasmolysis is deplasmolysis. Now tomorrow, children, I will be explaining you up to the experiment to demonstrate the process of plasmolysis and deplasmolysis in plant cells, though I had explained you with the figure, but in detail we'll do tomorrow. And then I will be taking significance of plasmolysis also, and we'll see if we can do passive and active transport also. This we will be doing tomorrow. Thank you, children.